So Dr. Davide Spadaro is an associate professor of plant pathology at the University of Torino, Italy, and researcher at Agrinova. His research spans from plant disease to sustainable crop production, including plant pathogen diagnostics and interactions, biological control, and food safety aspects of mycotoxin analysis. He is a visiting professor at the Thammasat University, Thailand, and at the University Edinburgh, UK. He is also a research fellow at the University of Lleida, Spain, and at the University of Bonn, Germany. He is also a member of the Council of the Italian Society of Plant Pathology, Accademia di Agricoltura di Torino, working group of post harvest and nuts of the Italian Society of Horticultural Science. He has worked on several European and national projects. He has served as a member of the organizing and scientific committee of 11 international congresses and served as a session chair at 6 international congresses. He has given lectures and talk at 12 international congresses. He has published over 600 research papers and author of 12 book chapters and 4 patents. His papers on international peer-reviewed journals have been cited over 3,000 times with H index of 30. Now, please join me in welcoming Dr. Davide Spadaro to deliver his presentation on molecular diagnostic and detection techniques for plant pathogens affecting fruits and vegetables. Davide, the floor is yours.
Thank you very much, Saravanan Kumar, for introducing me and mainly for having me today together with you. I'm really happy to have this opportunity, and it would have been really a nice environment if we could meet face to face. So staying in Italy and thinking about you in Trinidad is a really different environment. Anyway, we will try to cope with this situation. So today I will present our experience on the development of molecular methods for the detection of plant pathogens. I will now share my screen. Just a second. Okay. Okay. Yeah, just where I'm based. I'm based in Torino. Torino is a city of northwest Italy. It is on the border to France and close to Milano. We have a faculty which is dedicated to agriculture, where we have also several courses in forestry, in food science, and in biotechnology. I graduated in agricultural biotechnology, and I will present now the experience of my group in our work on the detection of plant pathogens. So first, just to have the framework, I would like to say that we are facing really huge changes in our environment, and these changes, which is the globalization of movements of people, of money, of goods. Also, it involves the movement of agricultural produce, and so also the movement of pathogens and pests. Just you can see from this graph how many new diseases are reported every year by CABI, and this just gives you an idea how much is becoming easier the introduction of new pathogens in the country. Even in Italy, every year we have several reports of new diseases coming from other continents, other countries. And another aspect which is changing a lot the scenario is the climate change. Here you can see just the UK during 2018, and in the other picture, the fires in Siberia this summer, which were something really new compared to the past. So we have a lot of phytopathological issues. I just report some of the issues we are having in Italy that were just introduced in the last few years. There is, for example, a new pathogen occurring on peaches, which is Monilinia fructicola. There is a new pathogen on citrus, which is Philosticta. Then we have a new powdery mildew on hazelnut, which is Erysife corillacearum, just arrived this year in 2020 from Turkey. Then we have also new Coletotricum and alternaria on apple. So every year, really, we are discovering new diseases. So it becomes really important to have good diagnostic tools ready to be used. And the diagnostic tools should have a lot of characteristics because they should be fast, they should be easy to be used, they should be possibly used also in field during the cropping season. They should be used also during the packing house, during the storage of the fruit and vegetables, during the transport until the supermarket chains. If possible, they should be easy to be managed, maybe portable. They also should target several pathogens, not only one, and so they should have this approach. And there is really a need from all these characteristics from diagnostic tools. So about the pathogens, we can find them in every environment. We can find them on the plant. There are pathogens which are soil-borne, so coming from the soil. There are other pathogens which are foliar. You can find them on the green tissues. The soil-borne pathogens can be found on the roots. Then we have also the post-harvest phase, the storage of the fruit, where we can have other diseases. And the pathogens can be transported by the seeds and the propagation material, which is a really important source. And several industries, seed companies, are really scared and worried about the introduction with their seeds of new pathogens. They can be transported by the air, or they can just be found into the soil. So we need different tools. Some tools should be targeted. Targeted means that they should be designed in order to recognize maybe one pathogen, while other tools maybe could be untargeted. So they just found what is in the environment. And among the untargeted methods, we have culturing and the use of microscopy. But these traditional techniques are really labor intensive. Several times they are slow. We should wait for several days. They depend on the expertise of the researcher, of the technician. And so sometimes there is also a lack of expertise. While if we move to targeted methods, in this case, we have mainly two categories, so the immunological and the molecular tools. In this case, they could be used even by technicians which do not have such an expertise. And today we will focus on the red one, so on the molecular tools. Some of them are targeted, like PCR, qPCR, LAMP, nanosensors, and lab-on-chips, while other tools can be untargeted. We have the high throughput sequencing, and also something which is in the middle, which is the microarray. In principle, it could be considered an untargeted method, but at the end, it depends on how many array you are using. So just, I would like not to skip completely the immunological tests, because I believe that in modern diagnostics of plant pathogens, they have a really important role. They are really relevant, in particular when we are working with viruses. Maybe they are a bit less interesting for bacteria, and in general, they are not efficient to work with fungi. But they can be precise. They are generally cheap. I would say the cost of one analysis is around $10 US. They can be fast, so you have the results just in a few minutes. And they are based on the use of antibodies. So I will not go into the details of this technique, but just I would like to remember that they can be really important. And there are mainly two applications which nowadays are widely used. One is the ELISA, enzyme-linked immunosorbent assay, and there are several types of ELISA tests. Normally, they should be used in laboratory conditions. We have just here some example of direct ELISA with the recognition between the antigen, which is our target pathogen, and then the antibodies, which is linked to an enzyme that, in presence of a substrate, will generate a change in the color, which is later measured by spectrophotometer, for example, or mainly also visually, so you can just see if there is a change of color. There is also the indirect ELISA, where you have two antibodies, one which is recognizing the target pathogen, and the second which is just a universal antibody able to recognize the primary antibodies. And then you have the double antibody sandwich, or the sandwich ELISA, which is typically like a sandwich because the antigen is between a capture antibody and the primary antibody. And last type is the competitive ELISA, where you have a change in the color, but in this case, you will see the opposite. So as soon as the concentration of the pathogen will increase, then you will see a reduction of the color in your wells in the plate. Okay, and the second example of immunological tests is the lateral flow devices. I would like to say that they are really interesting because they can be used by technicians in field for field application. They are not quantitative, but only qualitative. So they just give a yes, no answer to say if a pathogen is present in your field, but they are quite well developed for the detection of viruses. So what you basically see is just one lane, which is always present if the test has not expired, so it is still valid. It's control line, and then you have another line, which is the test line, and you see a change, an occurrence of the color just when you have the positive presence of the pathogen. So now, keeping in mind this, I would like to move to the molecular techniques. And I have introduced in this slide just what we can do with DNA, or in general with nucleic acids. We have 4 main activities that can be done. So we can restrict, cut, or fragment the DNA with restriction enzymes. This is one thing that we can do. The second is the use of PCR by amplifying the number of initial copies of our DNA in order to make it visible with naked eye in an electrophoresis. Another possibility is to exploit the hybridization characteristic of DNA, because as you know, DNA is made by 2 strands, a double strand, and so it's possible either to separate or to create a hybridization between two strands. And last, we can know the sequence of our DNA either by using traditional tools such as Sanger sequencing, or also the next generation sequencing tools. So there are a lot of features which make molecular techniques really feasible today, because they can be really fast. And we will see that we have nowadays some molecular tools that can give results just in 10, 15 minutes. So now we can have really fast results. They are really sensitive, so they can see until maybe one single molecule of DNA of the pathogen. So it's the most sensitive techniques. They are also really specific to differentiate not only the species, but also at a subspecific level. They can be used for unculturable pathogens, such as obligate parasites, powdery mildews, or downy mildews, rusts, etc. They can differentiate between really closely related pathogens, and they quantify the presence of the pathogen. So I would like today to have a quick overview about several techniques that nowadays are available by exploiting these 4 properties of nucleic acids. And the first I will start is in red, is PCR, because it's for sure the most important, the most widely used technology that has been exploited in the last years. I just really say 2 words. In the PCR, you have the amplification of your target DNA. So to have this reaction, you should include some primers that are used by the DNA polymerase, which is the enzyme, which is synthesizing a second strand based on the target DNA. The template DNA. You should also add the bricks for these reactions, which are the nucleotides, and then there is a buffer normally. There are 3 steps: denaturation, annealing, and extension at 3 different temperatures, and normally PCR is run in a thermal cycler, which is an instrument, and you generally have a number going from 30 to 40, 45, sometimes even more cycles, repeating these changes of temperatures. So now going to some of the application of PCR, one of the first I would like to focus is the PCR with universal primers. And when we do amplification with universal primers, the aim is to determine the species of our pathogen. In this case, I'm speaking about fungi or bacteria. And there are some barcode sequences, which are sequences present in all the species of a kingdom, and they can be used by sequencing this amplificate in order to understand and identify the species. In the case of fungi, ITS, it is a ribosomal region, has been selected, while for bacteria, it is the 16S. And these are widely used.
¿Tiene un huevo?
Okay. Yeah. Just why ITS has been used so much, because it is present in high number of copies in the haploid genome of a fungus, from 60 to 200 copies, because it has some conserved regions, and on the conserved region you can design or use universal primers, which are good for all the eukaryotes. But in the middle, the amplificate has a sequence which is normally species specific. So it's really important and useful to adopt together PCR and then sequencing in order to identify and also for taxonomical purposes and phylogenetic analysis. I would like to say that ITS is not really magic, so several times it doesn't work because sometimes you cannot differentiate between species of pathogens, as ITS in some cases is not so good, not so relevant. So there are other useful genes that can be used for fungal barcodes. One is the translation elongation factor, for example. It is used, for example, if you work with the Fusarium species into this genus. Beta-tubulin is another one, which is used for penicillium. Calmodulin, it has been used a lot for Aspergillus, for example. RPB2 also for Fusarium and Alternaria. COX-1 is used for oomycetes, agents of downy mildews. And then, for example, IGS is quite long, is 2,500 base pairs. It has been used inside some species such as Fusarium oxysporum. But even other genes could be used. And this is an example of Fusarium. Fusarium genus, inside you have over 100 described species belonging to 20 species complexes. So you can try to identify by using the famous Fusarium Laboratory Manual, which is a book written by Leslie and Summerall in 2006. You can use macromorphology plate in different standard media, but it is very complicated to understand and reach the species if you are not specialized. So in this case, for example, the translation and elongation factor is a very good tool when you want to understand the species and you are working with Fusarium. We published one paper in 2010 when we wanted to identify the agent of this disease in Italian rice paddies, fields. And in this case, for example, we were isolating a lot of Fusarium species, but by using this TEF gene, we could see that only one species, which is Fusarium fujikuroii, is able to cause the bacane, which is this elongation of the stem of rice. And similarly, on another crop, in this case, it's rocket, which is a salad, which is normally used in ready-to-eat packages. In this case, we knew that the agent of wilting was a Fusarium oxysporum, so we knew the species, but we could not understand the forma specialis. And we could use this IGS, which is more variable compared to ITS and could be used for sub-specific identification. Now, we don't have so much time, but I would like to say that as much as we are moving towards a precise identification, we are seeing that only one tool is not enough when we want to identify a fungal species. So now we speak about this polyphasic approach, which is recommended for identification of particular genera of pathogens. So for example, in the case of Aspergillus, you should put together 3 different techniques nowadays. And this is recommended also by different international organizations. So the morphological characterization together with the molecular identification by sequencing some genes, and maybe not only one gene is enough, and together by characterizing chemically the extrolytes which are released by the strain. These 3 methods together, microbiological, molecular, and chemical, can really help in understanding the species. Here is a work that we published 2 years ago on the identification of different species of yellow aspergilli, which are producing aflatoxins in chestnuts. And we used, in this case, for example, the macromorphology together with the sequencing, in particular, of the calmodulin gene, and then together also the production of electrolytes such as mycotoxins. And the same story is valid also, for example, for penicillium. Also for penicillium, nowadays they speak about using this polyphasic approach with the microbiological, molecular, and analytical chemistry approaches. For example, we wanted to characterize the penicillium species present on chestnuts, which is an important production in our country. And just by sequencing and using macromorphology and then extralyte produced, we could find 20 species. 2 of them are also new species. And so, for example, we have called one of these Penicillium taurinensis based on our town, our city. So I just want to say that only one approach, only the molecular, sometimes is not enough. So now, changing technology, I would like to say just 2 words about this technique, which is in the middle between amplification and sequencing, which is the SCAR. Because once you have the sequence of a pathogen, maybe it could become interesting to design primers on this sequence. And so these primers are called sequence characterized amplified region primers, so SCAR primers, and you can design species-specific primers. When you develop a specific PCR, then you don't need anymore to apply together the sequencing, so you can reduce the costs. But specific PCR can be used when you want to confirm the species that you are hypothesizing. And for example, we developed these primers for a pathogen on kiwi fruit. It was designed on the ITS region. And just another example is the design of species-specific primers for the black Aspergillus carbonarius, which is producing ochratoxin in grapevine and then also on wine. In this case, we design the primer on a gene which is involved in the biosynthesis of ochratoxin, which is one of the mycotoxins. So now, just changing example, speaking about seed-borne pathogens, I would like to say that there are several methodologies which are established by ISTA, the International Seed Testing Association. Most of these methods are really labor-intensive because, for example, you can plate your seeds on a PDA, on a medium, agarized medium, and then you just wait some days for the fungal mycelium development. Or you can put some seeds always in petri dish, but in this case you can have a blotter test on paper with water, and you can see the germination rate and the pathogens occurring together with the germination of the seeds. But these methods, as I said, they are sometimes really long, especially for the seed companies, because they would like to know if they have, for example, to apply seed dressing treatment to the seed. So one possibility also in this case is to design a species-specific PCR. And for example, for this Fusarium fujikuroii, which is, as I say, the agent of this disease on rice, the bacana, we designed species-specific primers on an edylation which was present only in Fusarium fujikuroii in the gene for this translation elongation factor for Fusarium fujikuroii. And from this endpoint PCR, we also designed a real-time PCR with cybergreen dye. But I will spend later a few words about the real-time PCR. Just another technique which is a bit in the middle between restriction and amplification is the PCR RFLP, or called also ARDA in some cases. So it's a technique where you first have amplification with universal primers through a normal PCR, but later, instead of sequencing, you follow with a restriction with enzymes. And these techniques can also be called, if applied to ITS, ARDRAS, which is acronym for Amplified Ribosomal DNA Restriction Analysis. And the aim is, in this case, to reduce the cost of the determination of the species when you are doing a screening work. So if you have, I don't know, 1,000 isolates and you need to know which species they are, then you cannot sequence all your isolates, but you could use this technology, which is based on first amplification and then cutting with restriction enzymes. And this is one example that we did for some black aspergilli, always on grapevine, which is a really important production for Italy, for Italian wine. Okay, last application about PCR is when you don't know the sequence, is to adopt some kind of random PCR. I would not like to spend too much about this because I believe that they are not really useful for the detection of pathogens, but they can be used when you want to characterize or study the genetic variability inside a population of microorganisms. And you could use the rapid random amplified polymorphic DNA technique, which is very old-fashioned nowadays. It has been designed over maybe 30 years ago, but it is based just on a single primer, low stringency condition, and high number of cycles. And here is a rapid done on different strains of Fusarium oxysporum. Rapid, I would say, is not very useful because it cannot be repeated from one day to another, and it cannot be reproduced from one laboratory to another. But sometimes it's really interesting because you can find some strain-specific bands, and if you just elute the bands and design and sequence this band, you can later design scar primers. So I would say that can be used as an initial screening when you want to later design some primers. Okay, so now I move to the qPCR. The qPCR is a technology which is really interesting. It is a quantitative PCR, also called real-time PCR, because it monitors the amplification of the targeted DNA over time. It's different from the classic endpoint PCR because you can really quantify the amount of DNA, and it can be non-specific. So by using, for example, the cyber green, which is an agent intercalating the DNA, or it can be specific for your species or your, I don't know, forma specialis, etc. Because in this case, you can use some probes. And a probe is normally tagged with a marker, a molecule that can fluoresce, release fluorescence. But until there is a reporter close to the quencher, then you cannot see the release of this fluorescence, while fluorescence is emitted only during the extension step. So it's a very good tool because it permits to read the fluorescence emission. And then at each cycle, you can measure the fluorescence change and this can be related with the standard curve to absolute quantification of your pathogen. So real-time PCR is really interesting because you can quantify, so know the exact amount of your pathogen. Okay, so I would say that it has several benefits because qPCR can eliminate the post-process analysis. So to run a gel, for example, and to observe then the result, it is specific because you can use the probes. It's really sensitive because you can find until 1 copy per reaction. It's generally quite fast. You can use even 1 hour nowadays. It's quantitative, reliable, really informative. You can also find the different pathogens at the same time, so it can be multiplexed. And also there are portable platforms. Just an example, as I am working on this Fusarium fusaricoroi on rice, we developed a few years ago, 3 years ago, a TACMAN qPCR specific for Fusarium fusaricoroi, and this is the validation on different varieties, and you can see in black line the amount of DNA that we could find into the roots and also the leaves of the plants. These 3 varieties are susceptible to Fusarium fusaricoroi, while the other 3 varieties are more resistant. And what was really interesting is that even in the tolerant or resistant varieties, you have some amount of this pathogen, which is not causing the disease anyway. And the method was also validated on seeds in order to quantify the amount of pathogen in naturally contaminated seeds. By knowing the amount of DNA, then you can understand the amount of the number of cells. And just another example of TACMAN qPCR development, in this case against Venturiae inequalis, which is the main agent of apple scab on apple, which is the most relevant, the key pathogen on apple. In this case, it's really important for the farmers to know the exact moment during the season when there is the primary infection. So when there is the release of the ascospores. So in this case, it's interesting to use the spore trap. The spore trap is a tool to monitor the volume of fungal spores into the air. And there are different types of spore traps. The traditional ones use an adhesive tape that later should be controlled under microscope to read the ascospore releases. Or there are other spore traps where you have you harvest and gather together the spores, but directly in Eppendorf tubes. So they are already ready for the molecular analysis. And what we did in this case is first to study the sequence, and we worked on a particular gene. We found a deletion on this gene, then we designed primers on this deletion. It was first an endpoint PCR, and then from this we also designed the probe, and we developed a TACMAN qPCR, which is really sensitive because it permits to find until 2 femtograms of DNA. So it's really a low amount, the lowest that we could find with molecular tools, and is really specific for the pathogen. Okay, so that's an overview about the qPCR. Then we have the LAMP. The LAMP technology is not so new, because also this technology started to be developed almost 20 years ago. Anyway, it's a really interesting technology that has been grown a lot in the last years. It's like the PCR, but in this case, we have an isothermal amplification. So it's like a PCR, but running at the same temperature, constant temperature. And for having a loop mediated isothermal amplification, you normally need to have 4 to 6 primers, so not 2 internal, 2 external, 2 loop primers targeting 6 to 8 regions. And the DNA polymerase is not the TAC, but it is coming from another bacillus, another bacterium, Bacillus thermophilus. It's a heat-tolerant bacterium, which has, besides the replication activity, also the strand displacement activity. So it is able to separate the 2 strands of the DNA. And so there is not anymore the need of this step at 94 degrees in order to have the separation of the 2 strands of the DNA. It's quite simple technology, quick, because you can get result in 30 minutes, even less, even 20 or even 15, 6 sometimes. It's specific and sensitive, quite robust and not so much costly. But also the most important aspect is that you can move it to the field directly. So by using this, for example, portable thermal cycler or thermic block, then you can do the analysis directly in field. And I don't have time to explain the principle working on the lamp assay, but just keep in mind that you should have 6 primers and this high strand displacement activity. There are also some limits, but I would say they are not the most important aspect, because the limits is that having 6 primers, maybe sometimes it's difficult to design them. Then you cannot multiplex, you cannot detect more than 1 pathogen at the same time, and you cannot use Tachman probe, for example. Okay, how can you see the result of your amplification? You can see it by turbidity, which is caused by the release of magnesium pyrophosphate as a byproduct of the amplification. Or you can add some intercalating agent like cybergreen, so you can see a color change. Or you can also observe it, for example, you can use other intercalating dyes. Potentially, the lamp assay, which is generally qualitative, can also become quantitative. So you could relate the fluorescence intensity or change of color with the amount of DNA initially present. So it could be called like a sort of real-time lamp. And we had one PhD student developing a lot of these lamp assays, because we have a close collaboration with a British company which is selling these kits. And so just I would like to show some example, because we developed a lamp assay for Fusarium oxysporum lactuce, which is a pathogen causing wilt on lettuce. And the development of the technology was both the design of the primers, but also the setup of the crude extraction. So you can use just some simple tubes to extract the DNA in field, just in 3 minutes, and then run the lamp. And you can reach really low infection rates and detection limits, both on the plants and on the seeds. So it's also interesting for seed companies. Similarly to this, we developed for rice another lamp for 2 pathogens. One is Fusarium fujikuroi, the other is Machina porteorize, which is the agent of blast, a foliar disease of rice. And also in this case, we worked by developing a crude extraction together with a lamp development. Then we also developed in another project the lamp for Venturia inequalis, the agent of apple scab. And in this case, it is for the detection of the pathogen in leaves and also on fruits, so in symptomatic tissues, but also in asymptomatic tissues, and also on spore traps. So there is a potential to use the spore trap together with the lamp assay in order to avoid the microscope observation. And the limit is 5 picograms of DNA, which is also a very low limit of detection. Another example is a post-harvest pathogen, 2 post-harvest pathogens, which are Monilinia, lax, and fructicola, and we developed the lamp for these 2 pathogens with a similar limit of detection and the crude extraction starting from the fruit tissues. And last example of lamp is about detecting, always in the post-harvest phase, but in this case on nuts, the presence of aflatoxigenic aspergillus. In this case, it was developed together with an Italian company, which is selling nut chocolate cream all around the world. Probably you know the Nutella trademark. And the idea is to detect early in the field or just at the harvest or post-harvest phase the presence of aspergillus, but not all the aspergillus, only the ones which are aflatoxigenic. So the primers for this assay were designed on a gene which is at the end of the biosynthesis of aflatoxin. Okay, so it was for lamp. Now moving, we move to another technology which is quite interesting, is the lab on chip. I have put lab on chip at the interface between amplification, sequencing, and restriction, because lab on chip, the concept is just to reduce all what you normally do in a wet lab to the nano scale by using the silicon technology, which is also used in the development of IT technology. So in this case, you have the design of your platform. There is also later the 3-dimension printing, and then you have your lab on chip or lock prepared. And what you can do is PCR by using principles of microfluidics together with the microarray, with restriction enzymes, cutting. So there are several applications. Just we worked about this with the engineer university in Torino, the polytechnics, and we adopted different technologies for DNA extraction in some chambers, coupled with dynamic PCR. So by using this small capillary where you have your fluid, your sample to be analyzed, and it's cycling between, for example, 3 different temperatures, so that you can reproduce really cycles of a PCR. And this is just one potential application where we developed a lab on chip, but for the detection of Aspergillus flavus in hazelnuts in this case. And we did this in this small support system, and we applied it to real-time PCR with the primers for real-time PCR, but also with the real-time lamp. And the results were really interesting with the real-time lamp. So there is also this potential to reduce to small scale the lamp assay. Then another technique is the microarray. In this case, it's using the hybridization power or potential of the DNA. I would say that there is a very old technique, which is the Southern blot, where you have the target DNA on your support, on your nitrocellulose, and then you have a probe which is flowing, which is in a liquid condition. In the microarray, you have the opposite concept. So you have a solid support with probe, but there is not only one probe, there are several probes. And then you have the flow of your sample, and there is an hybridization when there is a similarity between the DNA in your sample and the probes which are on the microarray. So it's potentially interesting because you can have quite a quick response, high specificity, you can have multiplexing and automation of the array. I would say that the first microarrays developed were not so much interesting, and also they were quite expensive, but nowadays we have, for example, one company, which is Alere, American company, which is developing these tubes where at the bottom of the tube you have a small microarray where you have spotted probes of DNA, and it permits you to detect several pathogens, up to 100 or more at the same time, and just by looking at the change in color into the spots just in your tube. And it's quite a small machinery that can be used on your lab bench. So it has quite potential, though it's not so much developed commercially until now. Then just 2 words about nanosensors. Nanosensors is another technology where you use some small-scale devices at the nanoscale to measure some physical quantities. And normally there is a biosensors, so there is an interaction, for example, between a probe and your DNA. So it's a hybridization system, or there is an interaction between antibodies and the protein of the pathogen. So this is an immunological interaction. There are different tools in order to convert this interaction into a signal that can be analyzed. So there could be electrical detection, optical detection of this interaction, but also mechanical detection. And with the engineers, we are working on these nano-mechanical cantilevers, which vibrates at a certain frequency. On the surface of these cantilevers, you can find some probes or some antibodies. And when there is a binding between the DNA or a protein, surface protein of the pathogen, then you have a change into the frequency of surface trace in your cantilever that you can measure. And also with this, we tried to develop a prototype for field application where we could use together the spore trap for the detection of Venturia inequalis on apple, coupled with a nanosensor. So it is an integration of this, I would say, nanotechnology and biotechnology techniques to field application. So we are running late. I am almost at the end, but I would like to spend a few minutes about another technology which has a really high potential, which is a high throughput sequencing. I would say that this is to understand the sequence, and as you know, the price of sequencing has become lower and lower. So the first human genome had a cost of 100 million euros, while now we are at less than 1 1,000 euros. So really the cost of sequencing per base has become very low. There is a potential application also to plant disease diagnostics, because we could use, for example, meta barcoding, which is amplicon-based high throughput sequencing, HTS, or we could simply sequence all the DNA of a sample. This is called shotgun metagenomics. And I would say that metagenomics in general includes both. So the meta barcoding, which is based on PCR, and the shotgun metagenomics. So in this case, just there are several steps by using metagenomics. So you first have a sample, you extract DNA, you run a PCR, for example, on the ITS for fungi. Then you sequence the whole sample, so all the microorganisms, all the fungi which are present in your sample. And then you have the sequence clustering. So you assign a name to all the taxonomical units, which in the case of fungi normally is the genus name. It's difficult sometimes to reach the species level. So about this meta barcoding based on PCR, I would say that there are several advantages, because from the bioinformatic point of view, we don't need so much power. It's not so much expensive, because nowadays one sample can cost like 40, 50 euros or dollars. But there are some issues. One issue is that it's difficult to understand the species, in particular for fungi, because operational taxonomic units, OTUs, are based on 97% similarity of the sequence. So you really underestimate the real biological differences occurring in your sample. And there are some tools, bioinformatic tools, to pass this issue. One is to use the exact sequence variants, which are consistent between different experiments. The other is to use, for example, oligotyping, which is a bioinformatic tool in order to exploit highly variable sites in your sequence. Then there is another issue, which is you can see in the last works that you can never find all the microbial diversity. Roughly, you can reach the 50% of your diversity. So if you want to improve the abundance and the representativeness of your sample, you could use more primers for the same locus. You could use degenerate primers or primers designed on more than one locus. And ITS, as we already said before, is not always the best gene for running a meta barcoding, because it's not very good, especially for ascomycetes, to go beyond the genus level. So there are some ideas nowadays to develop meta barcoding specific for the genera. So for example, for Fusarium based on TEF gene, for Penicillium and Aspergillus on beta tubulin, etc. Okay, then last is the possibility to really sequence all your sample without a PCR step in the middle, and this is called shotgun metagenomics. In this case, you sequence all your sample, then you have 2 steps of assembling and binning in order to rebuild the genome of your species, of the different microbial species occurring in your sample. And this, yes, has some pros, which are the high resolution level, but it's really expensive. So the cost is 10 times more than meta barcoding. every sample could cost $500 US, so it's not really feasible. But you can find, for example, in a strange symptomatic sample, maybe a new species, a new pathogen which was not detected before. So one of the main issues with working with shotgun metagenomics is the fact that databases do not have enough information. And so there are genomes deposit lacking, and also gene bank is not very well revised to work on whole genomics. Okay, last is what can be the applications of high throughput sequencing. You can use it for surveillance of pathogens and monitoring. You can use it for tracking non-harmful microorganisms, for example, to determine the origin of an outbreak. And this is already used in foodborne diseases, so in medicine. You can also use to identify new invasive pathogens. When you work on surveillance, I would like to stress the fact that we should have some baseline surveys. So you should know what is the situation of your region, of your country, if you want to apply high throughput sequencing for surveillance. Because if a new pathogen occurs, you don't know if it's really a new pathogen or already presented but not previously reported. And last, I'm really at the end, is just an application of this high throughput sequencing with meta barcoding in a rice paddy by using a spore trap. And we gather samples for 73 days during the cropping season from June to September. We sequenced all the samples by using a MySeq tool. What we could see during the season by the Shannon index is there is a reduction of the biodiversity throughout the season. So you start with more species and you finish with less biodiversity, microbial biodiversity. And then maybe it's not easy to see, but it was very interesting to see that during the season you have a change into the abundance of the different genera of fungi. So, for example, there is a reduction of Cladosporium of Alternaria. There is an increase of Myrothecium, which is related, for example, to climate change and increasing temperatures, average temperatures. And really, by using these bioinformatic tools, which is oligotyping, we could see that inside the Piricularia genus, we could see different trends for the different oligotypes. And every oligotype could be more or less similar to a species level or a sub-specific level. So there are oligotypes decreasing during the season, but other increasing, for example. And a qPCR permitted us to confirm the results of the analysis by demonstrating the reliability of this use of oligotypes for meta barcoding. So this is my last slides, and I would say that there are several directions for molecular techniques. So we are going towards the quantification of the sample through the qPCR or also the LAMP. We are developing field diagnostics, for example, with lateral flows in immunology and these LAMP assays. We are using multiplexing with the microarray. We are also going through surveillance of the environment with untargeted diagnostics with these metagenomics. We can use miniaturization with the nanosensors, and then we have also online diagnostics with these nanosensors together with the lab on chip block. Okay, so I would like to thank all the people that have been working to develop these techniques in my laboratory, and also some very good collaboration with Israel with Samir Grobi, Michael Wisniewski at USDA, Neil Bonham at Newcastle, Jenny Tomlinson at Thera UK, Optigen, the company for the lamp, and Carlo Ricciardi at the Polytechnics. And then I also would like to thank all of you for your patience and to listening to me. If I have sometimes, I know I was a bit longer, maybe I could answer some questions. Thank you.
Thank you very much, Davide. It was quite comprehensive and excellent presentation on range of techniques available for detection and diagnosis of plant pathogens. And I could see starting from the immunological techniques and also molecular techniques, and both based on targeted and untargeted approaches to detect those pathogens. And of course, I really appreciate your time and the effort taken to prepare this presentation and for enriching our knowledge on the molecular techniques available for detection of plant pathogens. Colleagues, participants, if you have any questions, you could raise your hand and you could ask to Davide. I see a question posted in chat box. I will read the question probably for facilitating this process. I see the question from Araya, and it was interesting to see that you report the limit of detection in qPCR assays as concentration of DNA. What do you use as standard for relative quantification in qPCR assays? How do you develop these standards?
Okay, yeah, for the qPCR, we face sometimes really big issues. So one of the first issue we are facing is that we hardly find samples in nature which are free from the pathogen. So what we are developing is that even when you don't have disease, if you don't have symptoms, a lot of samples still have some amounts of DNA, maybe of your pathogen, which is simply a saprophyte. But to start, when we want to validate, first we start with the DNA coming from pure culture from a single strain. Then after this step, we pass to naturally contaminated samples, and then we try to find samples which are free from the pathogen, and then we quantify the DNA by building a standard curve. By knowing the weight, the molecular weight of the pathogen, then you can deduct for the amount of DNA the number of cells which are present in the sample. So what we find is the amount of DNA, and then we could transfer this to the amount of cells, the number of cells or spores which are present.
Okay, so again, thank you, Davide. And is it reported as mass of DNA of the gene per mass of tissue?
Yes, yes. Yeah, normally we report as amount of DNA per plant tissue or seed tissue, depending on the type of test we are doing. Yes.
Okay, thank you. So what about the threshold cycles, or else just indicating based on the CT values, comparing the healthy samples as well as the infected samples?
Yes, when we have, for example, now we are developing a qPCR for a new pathogen on apple, which is called the Ramularia mali. And we have already scanned a lot of apple samples, but we have not found one without this Ramularia. But anyway, normally the CT value in a healthy sample is going from 35 to 38 to 39, while the CT value for the disease sample normally is between 22, 24 until 28, more or less. So you can see that there is a huge difference, but still it's not a really free sample, what we would like for the validation of the assay.
Okay, thank you, Davide. Colleagues, if you have no more questions, but still Davide will be with us at the end of this webinar, because when we have the discussion at around 2 o'clock, and he will be joining with us. And also if you have any questions, and we can always post those questions to Davide, and we can have a discussion. Once again, thank you very much, Davide, for your time and excellent presentation. Grazie mille.
Thank you very much for inviting me. I will be available later.
Okay, great.
Okay.
Thank you.
Bye.